blood group system. Oh. So your anagram, do y'all have your anagram that I printed out?
The thing you gave us yesterday?
Yeah, with all the little plus signs and minus signs on it. Yes, so the RH blood group system is gonna be, always gonna be the first column on one of those anagrams, or the first section. And so this first section is the RH antibodies. That's the only blood group that we're talking about right now. So each one of these dark lines that goes down the middle separates the blood groups. So the main ones that we're gonna worry about is D, big C, little c, big E, little e, and then CW. If you look at, so this is the patient's antigen profile, the donor, whoever donated each one of these cells to be used to make this reagent. So if you look at CW, if you follow the line all the way down in that row, there's one cell on that whole panel that's positive for CW antigen. So what do you think that means for transfusion purposes? There's not many people that are gonna be exposed to CW if 1 out of 10 people has it on their cells. And most of these, I'm surprised that it's even on here, most of the ones in the hospital, you'll notice they don't even have a CW positive cell. So it's a low frequency antigen. Not many people have it, which means not many people are gonna get exposed to it to be able to make an antibody. So we're not gonna talk about CW very much. What else am I gonna tell you about this? Okay, and the RHs, something that I didn't know until I had been in the blood bank for a couple of years, just because I never thought about it, I guess. Somebody told me. So the RH group can show dosage, right? That's one of the ones that show dosage. So you wanna use the strongest antigens possible to try to pick up the weakest antibodies, because homozygous reacts stronger with antibodies than the heterozygous in somebody that shows dosage. So that's just something you have to remember. All of the cells in the RHs will have at least one that is homozygous. So on the screen, did I give you a screen cell, the one with just 3 cells? I copy that one yet? No. So you'll notice on the antibody screen, there's only 3 of them. All of the RHs are going to be homozygous. There's not going to be any heterozygous, because the antibody screen is your first chance to detect is one there. And so you have to, the FDA makes them make it that way. So it's going to make more sense when we look at a screen, and I'll make copies of those too to look at. So the history of the RH system. In 1940, Levine and Stetson linked the RH system to the hemolytic disease of diabetes and newborn. And then Landsteiner and, I don't really know how to say it, blood bank guy says it, Wiener. Landsteiner and Wiener found that anti-RH agglutinated 85% of human red blood cells and 15% didn't. So that's where they came up with the percentage. 85% of people are RH positive, 15 are negative. So I would write that down. There's probably gonna be a question on the test about that, so. And so if you're looking at how much of the population has it, they call it the frequency when we're talking about blood bank antigens, blood group antigens. So the frequency of the D positive phenotype is 85% of the population. And so that's the people that are RH positive. And the frequency of D negative Rh negative. And so the Rh blood group system is the second most important blood group after ABO. Since the discovery of the D antigen in 1939, so a long time ago, they have discovered more than 50 Rh antigens. So we're really only worried about 5 of them. And our main concern is D, so it's not that bad if we don't have to look at a few of them. So if you think about it, the FDA said these are the ones that we have to worry about. So these are really, out of all of the ones that there are, so if there's 50 in the Rh group alone, then there's a bunch more, because all the ones in these groups, some of them, like the KEL group, there's probably more in that group than there are on here. So we only have to worry about these right here, most of the time. So Dave Steiner Jr. figured out that human anti-Rh was similar to guinea pig and rabbit antibodies when stimulated with rhesus monkey red cells. So they took red cells from this monkey, injected them into guinea pigs and rabbits, and they made an antibody that looked like the antibody that people were making when they had hemolytic disease of the fetus and newborn and stuff like that. So they were investigating, trying to figure out what was happening.
It almost seems like you would have to be like on drugs to even come up with
So,
an idea to try.
I'm sure they didn't just start with the guinea pig and the rabbit. It was probably brutal. Whatever I mean, they didn't just know, hey, that's the animal I'm gonna try. There's no telling what they did. I don't even know.
That's terrifying.
I know, right?
That's so sick individual.
Yeah. Okay, so both antibodies were later shown to be different, but the name Rh remained. So they thought that the antibodies were the same. Well, then they figured out later that they weren't. Well, the world had already started using the term Rh, and so instead of changing it, they just kept the name. So, these antibodies, these Rh antibodies, if you make an Rh antibody, they're IgG. So, you know, when we're doing A, B, and D in the tubes like we did yesterday, and it reacted at immediate spin. So if it's an IgG, usually those antibodies only show up in the indirect antiglobulin test. So after you incubate, because they're IgGs, right? IgMs are the only ones that we can see in the tube at a cold temperature. But the way they've made the reagent, it has IgG and IgM in it, so we can detect it at immediate spin because it's so important. And so if you had a patient that made an anti-D antibody from being exposed to red cells, it's only going to react at the indirect antiglobulin test, more than likely. And so the reagent is just made different so that it shows up at immediate spin. But if the patient has a real antibody, it's probably not going to be seen right away. You're going to have to carry it all the way through to be able to see it. And when we do an antibody panel, that's going to make more sense. And so you know how like the ABO antibodies, you make them automatically. You have to be exposed to the D antigen to make the D. You're not going to make that one automatically. So if you're Rh negative, you don't make an antibody to D unless you're exposed to red cells. So that's what makes this blood group different than the ABOs. So genetics, I know you're tired of talking about genetics.
I'm having fun.
Current theory says that two closely related or two closely linked genes control the expression of all Rh antigens. So they're codominant alleles. Almost all of the blood bank antigens are inherited in this manner. So the ABOs, remember we already said that, they're codominantly inherited. You need to know that for the test. So the RHD gene determines the expression of the D antigen, just D. RHCE determines the expression of the C's and the E's. So that makes sense, right? So if you just, don't worry about this part. So this one does the D, this one does C and E. So you'll need to know that for the test. And this, they're two closely linked genes, and these are the genes. So there could be a few questions from just this slide on the test. So if you're Rh negative, that means you got no genetic material inherited from the RHD gene from both parents. Because if you got just one D gene, then you're Rh positive. I'll just write that down. Y'all get sleepy already?
I've been sleepy since like 2000.
Yeah, I agree. I think the whole world is tired now. We're just tired. Means that, there is no genetic material inherited from the RHD gene remember, this is the only one that codes for D. From both parents. Write that down. So you get one set of those D, C, and E genes from one parent, one from the other. You have to get just one D to make the D antigen. So anybody that's Rh negative, which is 15% of people, right, that are Rh negative, they got two little Ds, which just means that they didn't get a D from both parents. Do you understand how the genetics works? So another way to look at it, it means they got two little D's, which equals no big D. So there's no such thing as a little D. My D's are ugly. That's just what it means.
Wait, say that again.
So if this person that's Rh negative, which is anybody in here that's Rh negative, you got, if you're truly Rh negative, you didn't get any D antigens. And so just as a place marker, we use the little d because that's where it would be. But there's no little d in real life. That just means you didn't get a d from either one of your parents, your mom or your dad.
So two tiny D's.
Means there's no d. So yeah, two little d's means no d. Because there's no, if you look at your anagram, there is no little d. There's just big D. You either got it or you didn't.
Yeah.
You can get one or you can get two. Even if you get one, you're Rh positive. If you got none, you're Rh negative. Does that make sense? So I don't want to confuse anybody. They used to say, or what they thought at first, because they're still discovering stuff today. They thought it was controlled by 3 genes, but it's not. They discovered that it's just 2, the RHD gene, RHCE gene. these are the common RH antigens. The ones that we're really worried about are these. So, if you had one of these also, and you were doing a transfusion, so you don't, if it's not on here, you're not gonna pick it up on here. So you would go and do your full, anybody that has an antibody has to have an AHG cross match, a full cross match, Coombs, cross match through Coombs, all those mean the same thing. That means a full cross match through AHG phase.
Are we not gonna use 6 and 7?
Well, here's C and E. So there could be a CE that goes together. So depending on where they are on the gene, so that gets weird. So that's probably stuff you'll do in an MLS program. We're not doing that. We're worried about what we're gonna see here. So, but if they had one of these, they had an antibody to one of these, you're not gonna catch it on here. So you're gonna figure out what their antibody is, find units, say they have a big C. You're gonna rule out everything else. So if you prove that it's C, you have to rule everything else out. You're doing good, you find a unit that's antigen negative for big C, cross match it, it's incompatible. Then it's probably something else. But without reagents and cells that have this on it, you can't keep testing. So what I do, this is not very common, but it can happen. So you only can test for what you have the ability to test for, which is what's on here, and that's it. In a regular transfusion service, because you're not gonna be in a reference lab. But there are some people that graduated from this program that work in a reference lab now for the Red Cross, and it's super cool. So maybe there you could do that kind of stuff. But if it was incompatible at AHG, you just get a different unit. So just find another one and cross match it and see if it's compatible. Because without the resources to figure out which one of these it is, there's nothing else you can do. So just get a different unit. So that's the main ones we're gonna worry about. So the Fisher-Race terminology, each gene expresses an antigen that's given the same letter as the italicized letter of the gene name. So those are not terrible. I'm not gonna really ask you that. So the C gene produces the C antigen. So that's easy, at least they make something cool. The absence of the D antigen is written as little d. So little d just means there's not a D. There is no real thing as little d. The order of the genes is usually D, C, E, which is how we did it yesterday. But it can be C, D, E. Why would they do that? D, C, E is how we're gonna do it. And that's how they do it on here. That's just how we're gonna do it. That's how we're gonna do it. That's just me. Blood bankers do weird stuff. So the Wiener terminology, according to Wiener, there's all these different alleles. Each gene encodes for an agglutinogen that's made up of these factors. This was the way they used to do it. It was called the longhand. We're not doing that. People don't do that anymore. Let's keep it as simple as possible. So the Wiener terminology shows you how to convert Wiener to Fisher. Fisher is what we did yesterday. So big R means big D, right? That's what we did yesterday. So that's why I wanted to do that yesterday, so you weren't looking at me like I had two heads today. Little r just means that there's not a D. 1 means the first letter is capitalized. So if we're doing it in order of DCE, which they didn't, it drives me crazy. But anyways, do it how, and so here, they did it here. Who did this? Do it the way I showed you. And so I got this one from the book, which both of those came from the book. This is the way that we're gonna do it to keep it simple. But this is exactly what I showed you yesterday. So you're just gonna have to memorize that chart. Rosenfield, I'm not gonna ask you that. But, this is the one that they made where they could use, like, they give, numbers, computers use numbers to code and stuff. I'm not a computer guru, but they had to make it where they could, like, where it would work in a computer terminology. So that's all that is. Rosenfield is what they do with computers. So if you went into like a LIS, like a laboratory information specialist position, you would learn how to do that kind of stuff. ISBT is the International Society of Blood Transfusion. So they came along, so you saw all those different terminologies and different ways to do the blood groups, antigens. So they came along and attempted to standardize it. Like we all have to do it just one way, or, because we're all studying how to do it, right? They had all these different countries trying to figure out how to call these blood groups what they are when they discovered them, and everybody was doing it a different way, and now we're crazy, right? This is just too much. So they tried to standardize it. So for computer sake, they had given us, each antigen gets a 6-digit number. And so, for example, the C antigen, which is the RH2, so they have the big C, that's this number right here. You don't have to know that. But like on units of blood, when you scan them in your computer system, when they come in at the hospital, that's what it's doing. It takes that barcode and gives it a number. That's how it does it. Okay, so the phenotype is the reaction between the red cells and the antisera. So phenotype, remember, that's what you can see in the tube. So if somebody was A positive yesterday and you saw the agglutination with the blue antisera, the anti-A, you can see that in the tube. That's their phenotype. You can't tell their genotype from that because they got gene from their mom and a gene from their dad. So they could have got an A and an O. You don't know. We just know what they look like in the tube. Genotype is the genetic makeup and can be predicted by using the phenotype and considering the race of the individual. So there are some blood group antigens that are more common in Caucasians, some that are more common in Black people. And then it can go way out into Indian people, Pacific Islanders, like, but we're worried about the majority of people that live here, right? And you have to do family studies or molecular testing. They usually just do molecular testing. They send it to the reference lab if they really need to know. That's the only way to determine their genotype. So genotypes can be predicted by knowing the patient's race. Common genotypes in Caucasians are the R1, little r, R2, and RO or R0. And in Blacks, it's RO, little r, R1, and R2. These are rare. And so I have a way to show you. There are some questions on this test about picking out which is the most likely antibody the patient could have if they had these certain genotypes. Or like, I'm not going to do that, but I want you to be familiar with how to do it. And so the blood bank guy has an easy way to show you. And it's on that video. I didn't post it yesterday because I forgot, but you should watch it. So the big four. So he did a box. And so we did whites and blacks. And so this is important, knowing the patient's race helps you determine what antibodies they could likely make. Some groups of people are more likely to make certain antibodies than others, depending on which genes they have on their red cells. And so the most common ones in both groups is R1, R2, that's not a Z, that's a 2. RO, and little r. And so remember, most people, 85% of people are Rh positive. So you have more big R's than little r's. 15% of people are Rh negative. So you just get one little R. So that's the most common ones in both groups. So if you start here and you start with RO, so in blacks, RO is the most common. And so you can go opposite in your other box over here. In whites, it's the least common. Box 2 is always little r in both spots. So we're just filling in the spots. This is something you're just gonna have to try to memorize by looking at it. So draw your little box over and over again and practice it with a blank box until you know, that's the only way you're gonna memorize it. And so now you just have to fill in the remaining blanks. So R1 comes first, so I'm gonna put an R1 right here. And then R1 goes over here. And then R2 is here. So R2 is all that's left. So it has to be here. I know that it's like, whoo, what are you doing? It's just something you have to memorize. So that blood bank video that I played the clip from yesterday when he was doing the nomenclature, he talks about this too. That's where I got this from. It's a very easy way to remember it. But it just takes time of looking at it and doing it to memorize it. Everybody write that down. So watch him do it too, because he's really good at explaining it. So these just shows you the phenotypes and genotypes, like are the most common ones. And here's their frequencies. I guess. Oh, there it is. The D antigen. So the D antigen is the most immunogenic in the Rh system. So we know that already, right? It's something we know. D negative people should receive D negative blood. D negative individuals receiving D positive blood can make an anti-D. So D is the most immunogenic, and that refers to its ability to make somebody make an antibody. As high as 75 to 80% of people that get exposed to the D antigen will make an antibody, is what they say, if they have a transfusion. So the more you get exposed to it, so if you have a baby, you're gonna get exposed to less blood than if you got a whole bag of blood, unless there was something traumatic happen, like a maternal fetal hemorrhage or something. So if you get a unit of D positive blood and you're Rh negative, you're probably gonna make an antibody. Now, what the blood bank guy said when I was listening to him is that the really, really sick people in the hospital whose immune systems are so busy trying to fix their problems that they already have, because nowadays with Medicare reimbursement and all that stuff, the only people that they're keeping in the hospital are the really sick ones. They just don't keep you as long as they used to. So it's not as high as they used to think it was, but it's really important for childbearing age females. Okay, the weak D antigens. So what it used to be called, and you'll probably see it in some of the books, it was called DU. And that stands for weak D. They don't use that anymore. We just call it weak D. And that's when red cells that test positive only by the indirect antiglobulin test are called weak D. So that's, you don't see it at immediate spin like we would see the ones yesterday. You only see it after you incubate, wash it, add that little ball of green reagent, the anti-IgG, you would add that, spin it down. That's the only way you can see it. Those people, even if you have to do that, they're still considered Rh positive if they're gonna be a blood donor. So the newer reagents have made it easier to detect the weak D antigens, but sometimes you still have to go through AHG to be able to see it. And so this might be a test question, so if you can only see it by the indirect antiglobulin test, that is considered a weak D antigen. Because it's weak, you have to do more testing to be able to detect it. That's probably the lab we're gonna do today. How many times did you get stuck yesterday? Twice?
Yeah, but it only,
If anybody if anybody that's D negative wants to donate some blood today, we can do a weak D lab, and so we'll do the washing, incubating, and all that stuff so you can see how to do it. I think that would be a good lab to do. So we'll talk about it after class.
just take 5 vials from me, it'll be fine.
So I kind of wish we would've took just more yesterday. The, freshman class are gonna start sticking today, and so I'm gonna get them to stick the blood, like draw blood bank tubes, and we'll save them, and I can type them and see what we have, if we have any successful sticks today. And I'm not gonna don't tell them we're using their blood, or we're gonna use it for blood bank. Okay, so, you have to do a control when you're doing the D test. If your control is positive, you cannot interpret the test. So if I give you some anti-D and control results on the test, the only thing you need to worry about is if there's a positive here, they're RH positive. But if this is positive, if your control is ever positive, you can't interpret it. And so literally in the computer, you would say unable to interpret without further testing. So that would be one of your options when I'm asking you what their D type is. You can't decide. So what would you do if you can't determine if they're really RH positive or negative? What kind of blood would you give them?
Negative.
RH negative, just to be safe. We're just doing the safest thing possible. So we're looking for is spontaneous agglutination. So remember when I said you had to do the D control, if your A, B, and D, all your forward testing is positive. So you don't have anything to prove that it's not just spontaneous, something that's making everything positive, like another antibody or Rulo or something, I don't know, anything, could be anything. So you do the control. If the control is positive, you can't interpret it. So this negative control proves that this is not just spontaneous agglutination. It's probably real. And these are both negative, so they're RH negatives. Negatives, negative. Okay, so the serologic, everything we're doing in blood bank and tubes, those are serologic testing. That's what it's called. This is something I want you to write down. The serologic weak D definition. Of red blood cells with an anti-D reagent. Weak. No or weak. And what I'm calling weak is less than or equal to 2+. So less than or equal to. Initial testing. Initial testing is immediate spin. That's what that IS means, immediate So that's what we did yesterday. We did an immediate spin D test. And moderate. Or stronger. Illumination with AHG or anti-IgG, that's the same thing. So you know how in our forward testing, we had 4+ A and B is what you're expecting. With the D, you should get at least 2+ if they're Rh positive. So remember, that one wasn't as strong as the A and B, that's normal. Now, if you get a 1+, you're like, maybe I need to do some more testing. So you would go through AHG and see if it gets stronger. If it gets stronger, if it turns into a 3 or 4+, they have a weak D, which would still make them be considered Rh positive. They can get Rh positive blood, but they can only donate blood to Rh negative, Rh positive people, because you can't take any chances of exposing somebody to the Rh antigen if they're Rh negative. Got it? Did everybody get that? Okay. So the weak D is more common in the Black population, and it's due to the inheritance of a weaker form of the D antigen. So often this haplotype, you don't have to know that. IAT is usually required to detect the weak antigen. So that's the indirect antiglobulin test where we incubate. We haven't done that yet, but once we do it, it's gonna make it click. We'll probably do it today. I wish I would've turned those heat blocks on this morning. Make sure when we get up there, we turn the heat blocks on so they can be warming up while we draw blood, if we draw blood. Patients are considered weak D positive, and they can get D positive blood. Why is the determination of the weak D antigen, or the D antigen, so important in pregnant people? Yes, HDFN. You don't want to expose them to the Rh antigen. So what can they do to prevent Rh negative people? What can they do to prevent immunization? RhoGAM.
Here's what I found.
So if kale is the second most immunogenic, we don't have, so this is kind of like, RhoGAM is kind of like a vaccine towards the Rh antigen. So they don't make one for kale. I don't know why they don't have one for kale yet. Kale is the second most immunogenic, but it's not really that big of a deal because so many people are kale negative that finding kale negative blood is easy, and so they really don't worry about it. Some people, some places, especially I think like other countries maybe, I've read in the blood bank group that they give, if a pregnant woman needs a transfusion, they just automatically give kale negative blood to keep them from making an antibody. Most places don't do that here because it's a lot of extra work. And the chances that they make an antibody, it's like, they'd rather just worry about it later. So, it doesn't really cause hemolytic disease as a fetus and newborn as bad as D does. D's the bad one. I mean, it will kill a baby. So that's why we do it. That may be a test question. The RhoGAM, the reason the importance of the D antigen is so important for pregnancy is because you want to keep the mom from making an anti-D. So the D antigen may appear weaker when the C antigen is inherited trans to D. I'm not going to ask you anything about that. Patients who are considered weak D positive are able to get Rh positive blood. This is the procedure for the weak D test. That says wheat, it's supposed to say wheat. But it tells you what to do it. So you'll get 2 tubes, one for your control and one for your patient. You'll put your anti-D and your control in there. So the control can just be saline, it can be albumin, whatever the procedure says. 1 drop of cell suspension, 2 to 5%. So remember, we always use 1 drop of cells suspension in each tube. to, you'll add anti-D to 2 drops. 1 drop,
One.
1 drop,
It's one.
1 drop. 1 drop, 7, 1 drop of the anti-D in the test tube, 1 drop in control tube, of the control tube. Incubate it 15 to 30 minutes, we do 15. Wash it 3 times, add 1 to 2 drops. Your procedures even in the hospital are gonna say 1 to 2 drops of anti-IgG. Everywhere I've seen them, they do 2 drops of the green liquid. Then you spin it down, and then look for agglutination.
So yesterday we did 1 drop.
Yeah, I don't know why I said that. 1 drop, I was thinking 2 drops of plasma. And those reagents are so strong, you only need 1 drop. So we use the anti-D reagent and 1 drop of cells, which is what we did yesterday. Sorry. That's why you follow your procedure. Don't just listen to me. Look at your procedure. So the reason we talk about this so much, they're Rh positive, they can get Rh positive blood. Nothing wrong with that. A partial D. So individuals that are partial D, they're D positive, but they are missing part of the D antigen. That's called a partial D antigen. When they get exposed to the whole D antigen, they can make an anti-D towards the missing part. So if you ever see a person that's Rh positive, but it looks like they have an anti-D, and you're like, hold on, that's not possible. Because if they're Rh positive, they can't make an antibody to what they have. Well, if they don't have the whole thing, they can make an antibody to the part that they don't have. I don't know why. I know, it's crazy. Partial D antigen reacts strongly with monoclonal reagents, and so you suspect a partial D when a D positive person looks like they have an anti-D. So they're not making an antibody to their own cells. They've gotten some D positive blood, and they made an antibody to the part that they didn't have. It's very annoying. I have not seen that very often though. That may be a test question. If somebody that's D positive looks like they have an anti-D, it's probably due to this one right here, a partial. So they make antibody to the part they don't have. So weak Ds can get Rh positive blood, no problem. Partial Ds can make an anti-D. It would still be hemolytic. If they made one, you'd have to give them Rh negative blood for the rest of their life. That's the worst thing that can happen. Okay, AABB requires that all donors be tested for the weak D antigen if they're initially non-reactive. So anybody that looks like they're Rh negative gets a weak D test. If they're donating blood, so that's a donor center, because they're distributing that blood to be transfused to other people. So if they look like they're Rh negative, they get a weak D. So if you're incubating, you do a weak D, it's negative. You have to incubate it, wash it 3 or 4 times, that adds 30 minutes onto your test. So testing for the D antigen, the weak D antigen on recipient samples is not required. So in the hospital, you're not gonna do that on every patient, because it doesn't matter. If they look Rh negative, you're gonna give them Rh negative blood and move on. So they may be weak D, but you're still gonna give them Rh negative blood, because that's the easy thing to do. Now, if they're sending blood out from the donor center to be transfused, you have to know for sure it's Rh positive or negative. That way you're not exposing somebody. Does that make sense? When I first started with the blood bank, we did a weak D on every person. So when it's positive, this just was very annoying. It added lots of extra time on to every test that I had to do. And back then, we didn't do it on the machine. Now they do it on the machine at most places. Well, I had to incubate it every time. It was so much extra work. So now, they don't do it on every patient. Doesn't matter. What they do it on, right there. RH negative mom. That gives birth to an RH negative looking baby. So that means the baby, if they appear RH negative, that means like if they look like they're RH negative at immediate spin or in the initial test, that baby gets a weak D test. What do you think?
It could be D positive.
To make sure that they're not RH positive and you're not exposing the mama. So this is to determine if she needs RhoGAM. That's what it all boils down to. So at around 28 weeks, during the pregnancy, you know, you're pregnant for about 40 weeks. If she's RH negative, she automatically gets a dose. So at 28 weeks, the baby's still in there. It's only like halfway cooked. We don't know. We don't know what their RH type is. There's no way to know. And it's too invasive to find out. It doesn't matter. We give her a prophylactic, is what it's called, dose of RhoGAM. So that's just in case. Her baby might be, oh my God, I'm sorry. Her baby might be RH positive, RH negative, but we don't know. And it's so important to make sure that we don't expose the mama, that we just give her one just in case. When the baby's born, she gets another shot. If the baby is RH positive or weak B positive. Because we consider either one of these RH positive. If the baby is truly RH negative through AHG, then she doesn't need one. Because there's a chance that if the mom is RH negative, there's a chance the baby could be truly RH negative. And so we wouldn't give her another dose. RhoGAM is a human blood product. You wouldn't want to get it, I mean, it's safe most of the time. If you read the package insert, it tells you that, and I've read the package insert when I've had trouble with babies. So you might not have access to it. At my hospital, we gave RhoGAM from the pharmacy. And in the doctor's office, when at their 28-week visit, the doctor gives them the RhoGAM. You don't even know that they did it. But there's side effects to everything. So it can be anaphylactic. Is that common? No. But it's still a drug. You don't want to give it to her if she doesn't need it. But if she does, we do, right? So if the RhoGAM covers 30 mils of whole blood, you don't have to write that down. We're going to talk about it in the baby chapter, mom and baby chapter. So there's a test that we do called a fetal cell screen after the baby's born. And it's just a tube test, and we're looking to see if mama cells has baby cells in it. We get a tube drawn from the mama after the baby's born and look for fetal cells in her blood. If there's more than a certain amount, because RhoGAM covers 30 mils of whole blood. How many mils are in those two that we've been using? 6? The tall tubes have like 6 mils? So however much 30 of those is, RhoGAM covers that much. So we will do it, if the fetal cell screen, where we're screening the mom's blood for the fetal cells, is positive, then we'll do a stain, put it on a slide. We'll make slides. That's the only slides we really make in blood bank. And we'll count the cells and do a calculation, which you'll have the formula. And that's how you decide if she needs more than one vial. So if she had more than a 30 mil bleed, then you give her, we'll calculate how many she really needs. And so if you dispense it in the blood bank, most of them have moved it to pharmacy because we have enough to deal with. We'll tell them how many vials she needs and they give them to her. And if you dispense it, then you'll allocate however many. It comes in a box. It's like a syringe. usually. So that's one of the fun things that we do in blood bank. So our job is really important. Any questions about that? Yeah, I'm not gonna ask you about that on the test.
The ROGAM shot at 28 weeks, and then the other one is after the week D test.
Yeah, after the baby's born. So if it
Okay.
looks like it's RH negative, they'll do a week D. If it's week D positive or RH positive, mama gets another shot. If her fetal cell screen is negative, she gets one just in case. And then if her fetal cell screen is positive, she'll get however many we tell them she needs, which can be up to like 5. You can only have 3 in 24 hours, I think. So they give her a couple, next day maybe give her another one. I can't remember the rules. But when I was having issues in the blood bank one day, I can't even remember what my problem was. I didn't have ROGAM in the blood bank, so I didn't have a package insert. So I Googled it. It's all online. So don't be scared to read the package insert. You find good information on there. So what I could ask you, so if a person appears to be RH positive, and they have an anti-D, that's to the partial D, y'all should have wrote that down. And then testing for the weak D expression is performed by the indirect antiglobulin test. Weak D, we take it through AHG, which is the indirect antiglobulin test. That's what might be on the test. Compound antigens, I'm not gonna ask you about that. And in my opinion, this is like MLS stuff, but an example of a compound antigen, when 2 genes are inherited on the same chromosome, so cis on the same, right? They can form additional antigen products. So somebody that has the little c and little e antigen can make a F antigen. So if somebody has an antibody to that, look at your anagram. Is F on there? So that, it would be in the RH group. So that's where you look. That's the only place you have to look. It's not on there. We're not worried about that. G antigen. So almost all genes that code for D or C will code for the G antigen. An anti-G antibody can look like an anti-D with an anti-C, big C. So if a person is negative for D and or C, and they get D negative, D positive or C positive blood, they may make an anti-G, which will look like they have a D and a C. So so you're in transfusion, you're really not gonna worry about it. So if the anti-G is present, you give D and C negative blood. So you would know what to do, because if they're D negative, you're giving them D negative blood. If you do an antibody workup, it looks like they have a C2, then you're just gonna give them D and C negative blood, and that covers it. So you're not gonna make a mistake because of the anti-G, because at least it looks like this, and given this is safe for this person.
So one adopted negative.
Yeah, so you'll know what to do. If it looks like a D and a C, then you're gonna give D and C negative blood, and then it's gonna be fine. Yeah. So G's not on here either. You're not gonna know it's a G. It's gonna look like a D and a C. And that's stuff that you're just gonna learn through experience. Okay, unusual phenotypes. The only one I'm really gonna talk about is this one. And I'm really not gonna talk much about it. So there, you can have no RH antigens on your red cells. So if you look at your anagram in the RH group, the plus sign means that that donor has that RH antigen on their red cells. If you're looking at it, there's not one on this whole panel that has somebody that's negative for everyone across the board, right? So if you look at it, they all have positive something. They have either a big E or a D or a little c something. So if you don't have any, like if you're negative through that whole part of the RH part, then they call that RH null. That means no RH antigens. So the lack of RH antigens on the red cell causes a membrane abnormality that shortens the red cell survival. So that's just something that you're born with. So if you ever hear about that, I don't ask you about that, but for your boards or whatever, now that you've heard of it, which blood group, RH blood group inheritance pattern can cause shortened life of the red cell? Null. No RH antibodies. Null means none. No RH antigens, sorry. Okay, RH antibody facts. They're usually, the antibodies, we're talking about the antibody. So we've been talking about the antigens before. They're usually red cell stimulated. So that's through transfusion, which is abbreviated TX, or pregnancy. Most are IgG. So you're doing the D test in the tube, in the forward, it reacts at immediate spin. You got a 5-minute test, we know they're RH positive. You're testing for the antigen when you're doing that. If you're testing somebody for an anti-D antibody, you're only gonna see it at AHG. So if you do just an immediate spin, you're gonna miss it if they have an antibody. That's why every antibody screen is carried through AHG, because these IgG antibodies are the most clinically significant. Except for the ABOs. Which are IgM. So you know that at this point. And so you can detect these at immediate spin, which is what the type of test we did yesterday. You have to incubate to find these. So IgG antibodies, so these are cold. They react at room temp or colder. These are warm, IgG's warm. So remember, that's 37 degrees, body temp. So you have to incubate, so you have to go through AHG to be able to see them. Potentiators are useful for identification. Somebody name one of them. Anybody remember one? There's like 4 of them we talked about. 2 that we use the most, like every day, kind of thing. Remember the one that Miralax is made out of?
Oh, yeah.
Polyethylene glycol. So nobody's gonna call it that in the blood bank. That is what it's called. So if you see it written out on a test, you have to know that that's what it is. It's PEG. We call it PEG. So PEG, LIS, those are potentiators. That just makes those IgG antibodies, because they're so small, come together easier. So antibodies to C, big C, little c, big E, and little e react stronger when they're homozygous, because they show dosage. And so on your antibody screen, all of the RHs that are on there will be homozygous. Every one of them, there's 3 of them usually. The one we're gonna use has 3. Every RH antigen that is present on the cell will be homozygous. So, don't forget that if somebody has a D antibody, it's IgG, because it's confusing since you're doing your IgM testing for the ABOs at room temp in the forward. That you're testing for the antigen. We're talking about the antibodies. They're IgG. That's what confuses people. So, the RH group shows dosage except for D. So it's part of the group, but it's different.
D does not show dosage.
No. So that's why it's listed here. These are the RH group. D is part of this group. But he don't hang out with them. He does not show dosage. It is the most immunogenic. So it's gonna be there. It's gonna be strong. These antibodies can go away over time. A D, unless the patient is super sick with no immune system, the D is not gonna go away, the D antibody. So D does not activate complement, the Rh antibodies, I'm so sorry I said that. Rh antibodies do not activate complement. And when they say that, they're talking about big C, little c, big E, and little e. There are reports of D activating complement. It's the most immunogenic. Doesn't mean it will, but it can. So it can kill you. So that just means that there are reports of it happening before. Doesn't happen every time, but it can. These do not. So these aren't gonna kill you unless you're already vaccinated or you have a really high titer. So big E negative and little c negative, so we're talking about the antigens, blood, that's sometimes given to when a patient has an anti-E, when the antibody is when the big E antibody is identified. So if somebody has a big E antibody, sometimes we'll just automatically give them C and E negative blood, little c, big E negative. And so big E and little c are friends. And so that's how I always remembered it. So I'm doing the antibody panel, and it looks like I have a big E. Big E is super easy to work up. That's everybody's favorite one to do, and it's like E for easy. It's easy to do. And then you see extra reactivity. I'm automatically going to look at little c first. Because if you make a big E, those people tend to make little c's over time, the more they're transfused. I don't know why, but it has to be maybe where the genes are. So that may be something that I ask you on the test. So if anti-E is identified, in a recently transfused person, which other RH antigen, AG stands for antigen, or I'm sorry, which other RH system antibody should be investigated? What do you guys say? Little c. Just remember, big E, little c are friends. Only two more. Sure. No, tell me, tell me. I'll tell me. Yeah, so big E and little c are friends. Okay. So the clinical consideration, we're talking about transfusion reactions. The Rh antibody levels may be undetected for years. So they make an antibody, maybe even through pregnancy. And typically people that are having babies are healthy. They're young. They don't have to go back to the hospital until they're in their 50s or 60s, say something happens to them, they get sick, or they get in a car wreck or something, they need some blood. They'll go to the hospital and they'll do their antibody workup and it's negative. So maybe they made a little E when they were pregnant. And then it's been so long since they've had an antibody screen, it's gone away. Because those antibodies, unless you're constantly stimulated, like our body, we're sick to something, we get constantly stimulated to the environment and all that stuff. Bugs, the stomach bug. hopefully we made antibodies to that. So you make these antibodies, and then you're not exposed to that blood again for, I don't know, ever, maybe. But if you do, and you made an antibody that your immune system recognized, they do your antibody screen at the hospital, and your antibody has gone away. Well, all you can detect is what shows up. So we call it negative, we give them a unit of blood, and they have an anti-little e. 98% of the population, I think, is little e positive. So the chances of you giving them blood that's positive for little e, it's gonna happen, it's probably gonna happen. Their immune system produces a rapid response that time. They start having a transfusion reaction. There was nothing you could have done about it because everything was negative. But their titer's gonna be high this time. So then, at that point, you just treat the transfusion reaction, and next time we'll know. And so that's why it's important to get the patient's history. Have you ever been transfused? If so, where? Some places, USA used to give them a card if they identified an antibody, and they put it in their wallet so they can give it to wherever they go. Hey, I have an antibody, because they can go away. Everybody doesn't do that. I thought we should have done that at the hospital. I think most of them probably don't. But do you think that's something that the patient needs to know? I think so.
Wait, they don't do the card anymore?
Some places don't.
Oh.
So the card would have to come from the blood bank. You would have to write it and put their name on it. I have this whatever antibody, and somehow get it to the patient, and so they just don't do it. I think they should. Because you go to the next hospital, and they don't know, and it goes away again 10 years later, well then, and you forget. Patients forget. They don't understand antibodies. They don't even know what an antibody is. So, I don't know, if I ruled the world, I would give them a card. Hey, you have this antibody. So anyways, sometimes there's nothing to do about it. Antigen negative red cells should be transfused if an Rh antibody is identified. So, we would give, if the patient had a little E antibody, we'd give them E negative blood. So if 99, say you have 20 units of, say they're an A positive person, you have 20 units of A positive on the shelf. You think if 99% of people are positive, or 98, I think it's 98, are positive for little e, are you gonna find a unit in those 20? Probably not. Probably gonna have to order it. And so that's what knowing the frequencies is good for, which in the blood bank, you'll have that little card and you'll slide it down and be like, okay, the patient has a little e, I need 1 or 2 units, and it's gonna tell you, you got to screen 100. That's how you know. That's the easy way. Otherwise, we have to do the math because we're in school, so we're being punished. So, I wouldn't even start screening. I would just order some blood from the blood center. All right, hemolytic disease of the fetus and newborn. It may occur when a woman is Rh negative and the infant is Rh positive. The antibodies are made in the first pregnancy. The baby is not affected because it takes a long time. So, within the maternal fetal blood exchange, they don't exchange a lot of blood. The baby makes its own blood and it does its own thing. Now, the blood exchange, if they exchange any, it's not much during the pregnancy, right? But when they're born, things happen. Birth is traumatic. So then that's when the blood backs up from the placenta, gets into the mama's blood. She makes an antibody for the next time. So that's how it works most of the time. So then the maternal antibody, the next time she gets pregnant, that's the baby that's affected. So as long as we give Rogam like we're supposed to, it doesn't really happen anymore. Now, there are still those weird people that think they know everything in vaccines and all this stuff, and they read the package insert, or I read about Rogam, and refuse it. There's nothing you can do about that either. That's the doctor's problem. So, in the modern day medicine, this should not happen with D. There's other antibodies that can cause hemolytic disease of the fetus and newborn, but they're not going to be as dangerous as D. If they have one that is causing a problem, they can do those intrauterine transfusions, because what it does is the mama's antibody attacks those baby's red cells, and so it's crossing the placenta and it's making the baby anemic. So it can take away from their oxygen carrying capacity. Sometimes they'll do intrauterine transfusions if it's really bad to try to save the baby. But that's not near as common as it used to be. And back then, when they didn't have intrauterine transfusion, the baby just died. And so that's why I use the Rogam for D. We only have in immunization for D, because that's the most immunogenic, it's the worst one. I've even heard of them giving Rogam to somebody that was Rh negative. Usually they wouldn't give it to a woman of childbearing age, but if there, say there was no blood available nowhere except for Rh, an O positive unit, and this O negative lady needed blood, and she was gonna die, they could give her, do a calculation, so a unit of packed red blood cells is 325 mils. If Rogam covers 30 mils of whole blood, you could do a calculation and try to give her enough Rogam to keep it from happening. If there was a complete blood shortage and she was gonna die without it, like, so I've even seen, or read, not seen, read